Thank you for saying all those words. I was challenged by Will's opening remarks. I searched for drug development jokes, and I got a question from Google, and the question was, do you mean drug development jobs? So I don't know if it's a joke to find a job these days, but we certainly cannot rely on Google to entertain ourselves. So what I was asked to do is to talk about something as fun and as intoxicating as chemical toxicity, more specifically chemical toxicity prediction, more specifically our recent work where we have challenged some of the long-standing approaches to toxicity prediction known as structural alerts, which is this word that I will ask you to pronounce slowly later. And to put sort of everybody on the same platform, let me tell you what's meant by this and make everybody a chemist instantly. I'm sure most people have dreamt about it since you graduated from the high school. So what's meant by this is something that is very simple and very desirable to have a simple tool to predict if a chemical is toxic or not. And that is to look at a bunch of chemicals that show toxicity in some tests or assays, and find out that they all share, or most of them share, a particular, rather simply looking chemical group, and then based on this analysis make a statement that all chemicals that have this group are toxic, such as, for instance, nitro-containing chemicals. And with that, well, the rest is a simple story. You publish, you get a grant, you become famous, get more grants, and then entertain yourself, well, publish software, sell it to FDA, and become really rich and really famous. And so that's a standard story as far as how things like this are developed, and it's easy to use, and it's something that we all want, simple tools to triage compounds that are unlikely to see the day of life because they will eventually be shown to be toxic. And the company, pharma company, chemical company profits will go down. So that's what it has been. Now let's look at this molecule, and some of you may recognize its structure and some may not, but it's the best-selling drugs of all time, and so it must be a good molecule. But then we also know about the history of structural alerts, and there is a lot of software that allows us to ask if such a group, or if a toxic group, or a toxicity or structural alert is present in any given molecule. And so you just ask a question with a lot of software that's written and available, if there is anything wrong with this molecule, and there is a reference if somebody wants to execute this yourself, and you find that it has idiosyncratic toxicity, and it has non-genotoxic carcinogenicity, and it has skin sensitization potential, and it could have genotoxic carcinogenicity, etc. So it has multiple functional groups that would make this molecule totally impossible to be pursued at the very early stages of drug discovery. And that certainly creates a question as to what is possibly wrong and what could possibly go wrong, and how come one of the most selling molecule that is still on the market could make it there, and why things are as bad as they appear to be. And then you look for what the developers of software to identify such alerts are thinking about when they run into multiple cases like this. And you find a code that I think is probably one of the best I've seen, and the developers are saying the name of the software that was called skin sensitization alerts is changed to skin sensitization reactivity domain to reflect the fact that alerts provide grouping into reactivity mode of action and do not predict skin sensitization potential. Now, what is something that is touted as predictive methodology or predictive software, but now is not used to predict what it's supposed to predict? So you search for answers, and I'm a chemist, but sometimes chemists need to go to quantum physicists for an answer. But before, you search for a pictogram that reflects this sort of attitude, and that's the best I could find. This is how I interpret the previous statement. So going to physicists, Werner Heisenberg once said there is a fundamental error in separating the parts from the whole, the mistake of atomizing what should not be atomized, unity and complementarity constitute reality. And that basically to say that sometimes the whole is more than the sum of its parts. And sometimes you cannot divide and conquer. Sometimes you conquer when you do not divide. And the examples are coming from the world of careful analysis of chemicals. And here is the nitro group. The nitro group that's supposed to be toxic all the time. And the reality is that if you analyze, so I'm glad you asked this question, because I have an answer. The reality is that whether or not nitro group causes toxicity, or mutagenicity in this case, depends on what it's bound to. So these examples, all of them, have this nitro group, but these molecules also contain an aromatic entity, and there is a mechanism explaining their toxicity, whereas when it's bound to an alkyl group, it does not cause toxicity. And that essentially says that what matters is the whole molecule, not any individual part. So is there a methodology that actually could look at the entire molecule? And that's a long-standing methodology. Thank you for the reminder. That's an alarm. That is called quantitative structure-activity relationship modeling that looks at the molecule as a whole, calculates something known as chemical descriptors, and then builds models that can be compared to, what else do I do? Is that's a dance of some sort. Maybe someone could push the button. Yeah. And what it does, it's a black box, that's the negative, but it does the same thing as the allures. That is that it predicts whether or not, but it does predict. It's not shy of saying, I predict whether a molecule is toxic or non-toxic as a whole without looking at individual composing allures. And so the next question, of course, is if you have alternative approaches to making predictions. One is based on something simple and transparent. And second, that's based on black box, which irritates every regulator and every biologist in the world. Which one would make more accurate predictions? So this is the experimental data about skin sensitization, and this is a comparison between allures-based system and whole molecule-based system. And of course, I chose the best example I could, but generally speaking, in multiple comparisons, this works about 50% of the time in a binary prediction experiment, and this works in about 70–75% of the time. And that's the result of multiple comparisons for the reason that the whole is not the same as the part. And so then the question is, what can we do now, and what's the final outcome of this type of analysis? And the outcome is rather simple. Can we use structural errors? Yes, we can. Can we rely on them? No. Can they work? Sometimes they can. Can we know if they work or not? Yes. If on top of creating a hypothesis based on the presence of certain structural alerts, we also use a holistic model that will evaluate whether or not this alert has a value quantitatively. And so we've come up then with a consolidating strategy that integrates the old rules and the old type of development model that, for the sake of being a black box, was not popular in toxicity prediction, and demonstrated that that gives us collectively combination of alerts plus alerts we could derive from these black box models gives us a much higher prediction accuracy in a number of toxicity cases. So that's pretty much what we've done recently, and I'll probably spare you of reading. I assume most people, I'm a foreigner, if I could read English, you should too. So thank you very much.